Hello, guys. In this short lecture, I'm going to tell you that why the B cell receptor, which is an immunoglobulin or antibody, is attached to a naive B cell, but later on, when the B cell is converted into a plasma cell after it has come in contact with some kind of antigen, then the plasma cells secrete the same immunoglobulin and it is not membrane bound. So, what is the difference at the genetic level when this antibody is processed? Um, you can see here that here is IgM, transmembrane IgM, and here on the right side is secreted form of the IgM. IgM normally is present on the B cell membrane. On the naive B cells, you would find an antibody which is IgM or IgD. So this transmembrane IgM, if you look at the genetic level, there is rearranged DNA. It means that I had already told you before that the gene segments that are going to code for the light chains and heavy chains of an antibody are present on different chromosomes. For instance, for the lambda light chain, it's on chromosome 22, and for the kappa chain, it's chromosome 2, and for the heavy chain, it's chromosome 14. And after that, somatic recombination takes place, and at the splice sites that I told you in the previous lecture, that different recombinases would recombine different gene segments with each other. So in this rearranged DNA of the heavy chain, you can see that the variable diversity in joining domains have been brought together as a result of somatic recombination. Similar is the case with these blue boxes. These are actually the constant region. And here is, in addition, is the MC region. The light blue color shows its MC region. So this is the region which is responsible for, adding a hydrophobic domain to this particular immunoglobulin or antibody. Uh, it is going to anchor the antibody or immunoglobulin into the membrane of the B cell. All the cell membranes, these are actually they have a hydrophobic interior because of the lipids that are present there, the phospholipids. All membranes are phospholipids actually. So their interior, the membrane's interior actually is hydrophobic. So this peptide actually, or this sequence that you see in the blue is coding for a hydrophobic, part of the peptide that is going to anchor this immunoglobulin IgM into the membrane of the B cell. So here you see that in the primary transcript, when it is transcribed and the primary transcript is formed, so this part, the MC part, is also transcribed here, followed by the poly A tail. And in the mature transcript, when the introns are removed from this primary transcript by different enzymes, and mature RNA is formed that also has this light blue MC sequences followed by the poly A tail. And when this is translated, mRNA is translated and the protein is formed here. This actually is the antibodies protein that has been formed. And along this heavy chain, at the end of the heavy chain, you see these anchor, which is shown in light blue color here. So these anchors actually are the hydrophobic domains of this heavy chain, which actually penetrates into the membrane of the B cells and keep this antibody is membrane bound. So it is attached to the surface of the B cell. On the other hand, in the secreted form of the IgM, when the B cell comes in contact with the antigen, and later on it is converted into a plasma B cell, then certain changes take place. For instance, you can see the rearrangement of the DNA as a result of somatic recombination. Again, giving you the same sequence for the heavy chain as you see in the transmembrane form. In the primary transcript, also you see that MC sequences in the blue color are present, but the poly A tail has been pointed to have shifted towards the right or the left of the MC sequences. Here you see the poly A tail in this primary transcript is towards the right, but here you can see it has been pointed here that it is towards the left of the MC sequences that are coding for the hydrophobic domain. And when the mature mRNA is formed, so this part actually is taken out or spliced out of this, all the introns are spliced out. So this is spliced out right, after the poly A tail, because poly A tail is the end of any RNA molecule. So as the MC sequences are lying beyond the poly A tail, they are taken out by different enzymes from this mature transcript. And you see that in this transcript, there is no MC sequences, unlike this one. So when there are no MC sequences, no hydrophobic domains are added to the heavy chains of the antibody, and that's why the antibody is not going to stay there on the surface of the B cell, and it is going to be secreted. This is secreted form of the antibody, and this can only be done, this kind of arrangement or rearrangement of the DNA is only done in the plasma B cell. And it does not require some kind of recombination. This is just alternative splicing mechanism that the mRNA, mature mRNA, when it is being formed, the primary RNA, transcript RNA is spliced at a different location in case of the transmembrane and in case of the secretory form of the IgM. Thank you.